It's a privilege for me to introduce Ned Sharpless. He is one of the leaders of our cancer center, the Lineberger. He's a Wellcome Professor at UNC. He's got all kinds of accolades. He's a wonderful geneticist. And I suspect that he's one of the major reasons I'm here, because when I was being recruited, or when I was auditioning to try to come to UNC, I met a lot of great people. I had some great friends here, Tom Shea, Lisa, Shelley, but I met Ned. And so for those who know me, I was actually silent for a half hour as Ned gave me a lecture on P16, genetics and molecular markers of aging. I even took about 2 pages of notes. And so I came home and... yeah. I said I came home and I told my wife, this is the place. So it's a privilege for me to introduce Dr. Sharpless, who's going to talk about controlling the cell cycle in cancer therapy.
In any event, my lab works on 2 kinases, 3 activators, and 4 inhibitors. So this kinases, CDK4 and 6, we've heard some about today, the D-type cyclins that activate them, and these inhibitors of those kinases called the INK proteins. And I'd initially planned to talk about both today, but Hai told me I only had 20 minutes instead of 40 minutes, so I'm going to talk about the aging part of it, senescence and aging today. So just the INK4 story. And INK4 is also, I'll call it P16 most of the talk. It's also known as CDKN2A, this locus that's been deleted on all these TCGA slides is being commonly mutated in cancer. So the relationship between senescence and aging is an interesting story that pertains to cancer biology. So by senescence, I mean this phenomena that Leonard Hayflick described in the 1960s when he was trying to make vaccines in cultured cells, and he noticed his cultured cells would stop growing after a while in culture, and he called that cellular senescence. And it's a very reproducible finding of primary cells. All primary cells pretty much will do it if they don't do even a premature senescence in culture. And it involves the expression of these markers, and the cells look different, and we're very good at identifying senescent cells. And for the purposes of today's talk, it's highly associated with this protein P16, which is a major effector of senescence. And I should say, when Dr. Hayflick discovered this, he made sort of 2 predictions that both turn out to be true. One is that cancers ought to never senesce. They ought to figure out a way to defeat this cellular senescence mechanism, and that was right. And they do it by deleting P16 and P53 and other mediators of senescence. And the other thing he predicted was that senescence, these cells running out of replicative capacity, ought to have something to do with human aging. And that turned out to be true too, but that took like 50 years to prove. So senescence is mostly caused by 2 proteins, the 2 pathways, if you will, the ARF-P53 pathway. P53 is a very famous tumor suppressor gene, and the P16-RB pathway, and RB is the first tumor suppressor gene discovered. And what's interesting about this is they're both encoded by this genetic locus, where 2 proteins that are very similar, P15 and P16, are right beside each other. And then there's a protein called ARF that's completely different. It shares exons with P16, but it splices into a different reading frame. So it's like a totally different protein. And what's funny about this is that all 3 of the proteins activate the senescence mechanism. So it seems to be a design flaw, if you will. The major effector of senescence in human cells is all put within about 30 kb of the genome. So that means that you can delete 30 kb of the genome and get an immortalized cell relatively easily, and that happens all the time. That's why 50 to some percentage of cancers delete this locus. It's the most commonly cited cytogenetic deletion in human malignancy. And the locuses have other names that CDKN2B and CDKN2A. I don't like this term at all because you could be talking about P16 or ARF and no one will ever know. They're totally different proteins, so I don't use it. And an experiment I did back when I was at Harvard as a postdoc was to make mice that couldn't do senescence. So I made animals that didn't have P16 and didn't have P53. And so I completely kiboshed the senescence mechanism of those cells. Those animals had no real cellular senescence capability. And the interesting finding about those mice is that they were pretty normal. They looked fine, they ran around the cage, they were indistinguishable from their litter mates until they all dropped dead of cancer in like 8 weeks. So they're very, very, very tumor-prone animals. And they would not develop one cancer, they develop like 10 at once, and they'd have multiple different histologies, and they're very tumor-prone animals. They die so quickly they didn't have time to breed. So this answered an important discussion that had been going on in evolutionary biology circles as to whether or not cancer was an evolutionarily protected phenotype, or rather tumor suppression. Many people said that cancer was just something that happened as an accident to old people, and it was stochastic in nature, and tumor suppression really wasn't important. But I think this proves that's wrong, that if we don't have tumor suppression, and we don't have cellular senescence specifically, then we'd all die of cancer very quickly. So those RAS mutations and those BRAF mutations that those guys talked about in that prior session, they happen all the time. They happen every day. They happen with every hour probably. So bon appetit. So not getting cancer is a criterion of successful aging. So right there's a relationship between cancer and aging that my lab has worked on for a long time. And we thought, well, what would be the problem of making a mouse that did too much tumor suppression? It would be easy to do. You could make mammals that didn't get cancer. They were entirely cancer resistant because of an enhanced senescence mechanism. And we hypothesized, and it turns out to be basically true, that the cost of that is premature aging. That the good news is the animals are tumor resistant, but the bad news is that the senescence mechanism causes normal cells even to wear out prematurely. And that decline of replicative function that occurs with aging is a characteristic thereof. And here's one example of that. These are cells in the brain that make new neurons in the mouse. These are neural stem cells. And they stop dividing, so that the green markers division, and this is where they're found in the subventricular zone of the brain, and they stop dividing as you get old. So that's one tissue where there's a dramatic decline in replicative capacity with aging, and we know a lot about this now. This is not unique to this tissue. T cells do it, pancreatic beta cells do it. There are many tissues in the human body that stop dividing, and that loss of regenerative capacity is why we get old, in part. So since the cell cycle, there's a lot known about it. It's known what makes cells divide and not divide. When that observation was first noticed, it was easy for my lab to rattle through all the things that regulate the cell cycle and see which of those changed with aging, and there are really only two. So this is a log scale on the y-axis. That means each unit change is a twofold increase in the expression of these various cell cycle inhibitors in old versus young mice. So you can see P16 and ARF to a lesser extent both go up sort of 10 to 100-fold with aging in a variety of tissues. And none of the other things that cause cells to not divide change. So if you think that a failure of replicative capacity is an important part of aging, which was controversial when we discovered this in 2004, but is no longer controversial at all now. But if you think a failure of replicative capacity is important to aging, these are the best two candidates of what are causing that. So it's not just that P16 is a marker of aging, we think it actually in some ways causes that replicative loss. And we proved that in mice in beta cells, so we made mice that have too much P16 and their beta cells don't divide. This is a marker of proliferation. So their beta cells don't divide very well when they're young. And then we made mice that have no P16 and their beta cells divide just fine when they're old. But this is not a good thing to be P16 deficient. These animals are resistant to diabetes. They have enhanced beta cell function throughout life. That's the good news. The bad news is they all die of cancer at about 1.5 years, right? So the trade-offs are very explicit. Cancer and aging, the hypothesis. Right. So this is sort of the model now that's become prevalent of sort of the senescence model of aging, if you will, that when you're young, you have these stem cells that work well. They make a tissue. When you're old, they don't work so well because they become damaged. And when they can't self-renew, they either die, and nobody knows what that does in terms of aging. That may be a good thing or a bad thing. They become malignant. They can transform, and that's what happens when you get these fixed mutations like RAS and P16 loss. Or lastly, they can senesce, and we think that's very promoting of aging in ways. So that's the model, and that's sort of the basis of my talk that now I'll launch into the clinical portion. So senescence and aging, this model makes sort of two predictions that you would care about if you were like a person or even maybe an oncologist. So prediction one is that since all people are heterogeneous, we're an outbred population, we share different genetics at all loci, that there ought to be people who activate these senescent set points differently from other people, and they ought to get different diseases based on that phenomena, right? My lab set about to do experiments to explicitly test this in humans back in about, I'd say 2006, when this model was sort of becoming prevalent. And we got scooped like no one's ever been scooped before. I mean, we got scooped by like 2 million patients of data because the GWAS revolution happened. And the GWAS, the genome-wide association studies, started mapping traits of human diseases to various loci. So here's one of the larger GWAS that's ever been published. This is something like 40,000 patients from the Wellcome Trust. And they mapped, each dot is a SNP, a single nucleotide polymorphism, ordered by chromosome in the human genome. And you see none of the SNPs really rise to the level of genome-wide significance, which is considered an odds ratio of 10 to the 6th, except for one locus, which is the 9p21 locus. And this is for heart disease. So, there was probably no one more interested in aging in P16 in the world than me at this time. And I wouldn't have predicted heart disease to be a phenotype associated with this. I would have thought cancer, maybe diabetes, stuff like that. But this is a very strange finding. And it's not unique to this disease. It's unique to like 10 different aging-associated diseases. So there's strong human genetic data from millions of patients that we age differently because of polymorphisms of this locus. And the full Molecular details of that are not completely understood, but I can tell you some of these SNPs certainly increase the levels of P16 and ARF. And so we decided, actually motivated by this, we just decided to look. We downloaded all the GWASs, they're at a database you can find, and we just lined them all up and said, where are the SNPs that are associated with any disease? We don't care what disease, as long as it's a disease. We threw out things for like red hair and cleft palate and stuff like that. That congenital conditions and traits we didn't include. We just included diseases. Ellen de Graffenried is laughing because some of us consider red hair a disease. But we threw that one out. And we were left with a bunch of disease GWASs, and we just said, where are all the SNPs mapped? And we lined them up and hit PITs per bin, SNPs per bin, and then we said, what are the hotspot loci for human multi-disease association? So in other words, human wellness. What are the genetics of human wellness, not getting a disease? And the answer is right here. The number one locus for human disease association is the MHC locus. This encodes about 800 genes involved in immunity. Not surprising, the immune response is very important to whether or not you get scleroderma or rheumatoid arthritis, pneumonia, stuff like that. The number two locus is the Ingarf locus, and these are really the only two that are statistically significant at a genome-wide level. So this led to my crazy Joe hypothesis, right? Now we all have a crazy Joe who smokes like a banshee, drinks, he has no good living habits, he's obese, and he lives to 104, and he never gets sick a day of in his life. So I think this argues the genetics of Uncle Joe may be relatively simple. There are a few loci that seem to be largely associated with wellness in large million patient, this is 2 million patients of data. So that's the set point thing, right? I think the data are reasonably convincing that the senescence locus is associated with how we age, age-associated diseases. The second prediction is that if P16 goes up and causes aging, then you ought to be able to measure P16 in some cell and decide how old someone is in molecular terms. So the molecular basis of aging should be easily testable. So we decided to try this, and we started this in about 2004, right after the mouse experiments I showed, and it took us till about 2007 to get it to work because we tried the wrong cells and we didn't have the right technology. And eventually the problem was solved by a postdoc in my lab named Yan Liu working with one of our new faculty, Hannah Sanoff. And together they developed this interesting approach where we would sort T cells from the peripheral blood, because there are a lot of T cells in the peripheral blood, and T cells express a lot of P16. And we would then do a questionnaire about people, and I'll tell you right away that we did not ask enough questions because we didn't expect the trial to work. So there are a lot of things I wish we'd asked, but we didn't. But we asked about, like, how much do you jog and smoke and stuff like that. And then we took some blood and we isolated the T cells, CD3 positive cells, and we made RNA because that's how you measure P16. And then we did a bunch of other stuff on the serum and the protein and whatnot I won't go into. And we found this out, that molecular aging is exponential. So on this x-axis is the age of the person, and the y-axis, again, a log2 scale, so every 1 unit increase is a doubling of the protein, is P16 levels. So in a general healthy population, you had to say you were healthy to be in our trial. It sort of goes up about 1.4-fold per decade. But you can see there are some people that are quite molecularly young, even though they're older, and there are some people who are molecularly old, even though they're younger. And we know some of the things that affect this. So one of the things that makes you molecularly old for age is smoking. Smoking is very bad for your molecular age. In fact, it's a much better gerontogen than it is a carcinogen because it promotes aging in everyone. Only some people who smoke get cancer, but pretty much everyone seems to have accelerated aging from it. We know exercise is surprisingly good. The effect of exercise was strong in a lot of different populations and reproducible. And no matter how we asked about exercise, it always seemed to work. And it wasn't just that people who exercise don't smoke, because in North Carolina, you can test that explicitly. Plenty of people who do both, and yet we could show in multivariate analysis they're separate. BMI wasn't bad, so being obese wasn't terrible for your P16. And we've now actually reproduced this in mice. We've made mice super fat. And their P16 does not go up, no matter. We fed them stuff that, yeah, I mean, my 15-year-old son wouldn't eat, and it doesn't affect their P16. So body mass index, at least, doesn't seem to be a major determinant of this kind of molecular age, whereas smoking and exercise are. Exercise good, smoking bad. And so you see, this is there's another marker for molecular age is telomere length, which we've used quite a lot and done a lot with. Interesting marker. One technical difference is important is telomeres change by about 30% throughout the human lifespan. That makes them rather difficult to measure reproducibly. This goes up 8-fold, right? So it's a very simple test to do. It's a log scale, remember. So we think it's a robust marker of aging. So when we had this invented in 2007, what could we do with it? Being an oncologist, I think there are a lot of things one can do with a molecular test for age. But being an oncologist, I had my obvious bias towards cancer. And so in 2008, Hannah and I, and later Hymus, started a study to investigate the role of molecular age, or P16 as a test of molecular age, and a variety of cancer outcomes. And since Hy arrived, there's been this sort of onslaught of trials now, all of which that Hy and others that he works with have started. And two of them are complete now. And this one's even been published in an interim analysis at ASCO as an abstract. But I'll talk about this one today that is not published, that is 0810, which is a trial that works like this. You have breast cancer, you're going to get adjuvant chemotherapy, and we required that that chemotherapy be 4AC or rougher. So that's anthracycline, Cytoxan, adriamycin, Cytoxan. And they could get dose-dense therapy, they could get AC Taxol, they could get some variant of AC, but it had to be at least AC or worse from the side effect point of view. And then we followed them, and we measured P16 before, and that's the pre-value. And then we measured P16 at 3 times points after the 1 month, 3 month, and 12 month. I won't show any data for the post values other than I'm just going to group them together because they're all the same. Basically, your P16 goes from lower to higher, and it stays high for a year later. So we did this test, and then and here's the answer. Each line is a patient matched with their pre and their post, and you can see just about everybody goes up. One person goes down, and that person is interesting. That patient had Li-Fraumeni syndrome and died shortly after. So a germline P53 mutant patient. Very strange. I know why their P16 is high to begin with. I don't know why it goes down. I can't explain that. But Li-Fraumeni syndrome is a DNA repair deficiency syndrome. Failure of DNA repair causes higher P16. So this part's very easy to explain. So everybody goes up, and the magnitude of the effect I think is remarkable. On average, it's about 1 log. That's twofold. It's 1.4-fold per decade, so that's 2 decades of aging. Your bone marrow, not all of you, but your hematopoietic compartment is rendered about 20 years older from commonly given cytotoxic chemotherapy for breast cancer. Now, this is the part that's research for me. They don't let me speak to clinical audiences very much, and I think probably by now it's for obvious reasons. How many people in here make a living giving chemotherapy, cytotoxic chemotherapy on a regular basis? Raise your hands, everybody, right. A lot of you. How many of you, keep them up. Hi. How many of you are surprised by this? Nobody, right? I mean, we all know that chemotherapy makes our patients somewhat more fragile. And the more chemotherapy we give, we all know at some point they get worn out, they get exhausted, we can't treat them anymore. This is a fact I think every oncologist knows, but now we can quantify it. And I think this is and I think what I'm particularly interested in, by the way, is why do some people do this and other people do this? What's different about this guy and this guy, who had a 16-fold increase versus a 50% increase? I think that's really interesting. I can tell you from the other study we've done, I won't show any data, that the agent matters. We think anthracyclines are particularly bad. We think taxanes are not so bad. So we think that some agents are more age-promoting than others, if you will. So in summary, activation of the INCARFL tumor suppressor locus prevents cancer but causes molecular aging. People differ in their propensity to activate the INCARFL locus, explaining genetic differences in susceptibility to multiple diseases. And cytotoxic chemotherapy accelerates hematopoietic aging by about 2 decades. And these are the people that helped do the work, and I won't name anyone because I think I pointed out, and it'd be, well, I think there's no Q&A, right? So I'll turn it over to Hy.